big old luster balls.
It's a little dark, huh? Oh, is this okay?
No
Everyone has to-
PowerPoint. You gotta write everything.
So let's get started again. What are we doing? I don't know. We're talking about the regulation of microfilament synthesis, right? And let me go back here where no, that's fine. Right. So right off the bat, in order to regulate synthesis of microfilaments, the process of nucleation has to occur first, right? And nucleation can occur either with the help of formant or ARP. Okay, so that's that. And then we talked about thymosin and profilin, where profilin binds to the ATP bound form of G-actin to increase the rate of assembly, right? So normally, if G-actin were bound to ATP, then the G-actin can be used for assembly anyway. However, the rate is really slow. Okay, to greatly increase that rate, then profilin can bind to G-actin. All right. At the same time, thymosin can compete with profilin to bind to the ATP-bound form of G-actin, and thymosin actually will inhibit assembly. Okay, and in order to regulate assembly, therefore, the expression of thymosin and profilin has to be regulated. If you have more profilin, then that leads to assembly. If you have more thymosin, then that leads to the inhibition of assembly. Does that mean that disassembly occurs? Well, other things will regulate that. Okay, so again, having a higher amount of thymosin only inhibit assembly, not necessarily inducing disassembly. Be real careful on that. Okay. So next, how else can assembly and disassembly of microfilament can be regulated? There is this protein called a capping protein. Okay. It's not a cap like the cap on at the 5' end of mRNA. That's different, right? That's actually a nucleotide. This is an actual protein. And so a cap can be placed on the plus end, only the plus end of the microfilament, and that actually stabilizes the microfilament. And if that same microfilament, the nucleation of that microfilament were carried out by ARP, and guess what? Both the minus end and the plus end will be capped. That means that the microfilament will no longer be dynamic, and it will be stable. Okay? So a capping protein added to the plus end of the microfilament will stabilize the microfilament. What does that mean in terms of assembly and disassembly? The capping protein essentially will cancel out, will inhibit both assembly. Well, there are two types of disassembly, so I'll mention the words now, and then we'll come back to explain it later in a later slide. The capping protein will prevent both polymerization and depolymerization. Okay, and I'll explain polymerization, depolymerization, which is slightly different from talking about assembly and disassembly. Okay, so again, a capping protein will prevent both polymerization and depolymerization. And by doing this, the microfilament will now be stable. Okay? Don't worry too much about the graph here. Some more before we go on is, again, I keep looking at the next couple of slides, and they seem to be out of place, but that's where we put them. So in terms of bundling, right, how bundling or the aggregation of microfilaments are controlled is by microfilament associated proteins. So in order to form a contractile bundle, then there are these associated proteins called alpha-actinin, and you notice that alpha-actinin is a lot larger than fibrin, And that's why the contractile ring have a lot more space in between the microfilament and than the tight bundling, right? You don't have to memorize alpha-actinin and fibrin. Just do realize that the bundling, right, the three forms of bundling or aggregation of microfilaments depend on microfilament-associated proteins. So therefore, the gel-like structure also depends on microfilament-associated proteins. In this case, it's filament, but again, don't try to memorize these names, right? It's not necessary. Just know that microfilament-associated proteins are involved in bundling or aggregation. Yes, ma'am.
Is there different types of microfilament-associated proteins or different types of bundling?
Yes. For instance, for gel-like structure is filament. For contractile bundling is alpha-actinin, and for tight bundling is fibrin. So absolutely. Okay. So more regulation. Oh, not yet. Microvilli. Remember, microvilli is an extension of the plasma membrane, and is produced by not only the assembly of microfilament, but they are tightly bundled. Okay. Let me try to remind myself what's next. Okay. So just since let's see. Yeah. Okay. So microvilli. Anybody know what the function of microvilli?
Increase the surface area, right, for absorption?
Not absorption.
No.
Increasing surface area, right?
Yeah.
So in microvilli is where a form when the assembly of microfilaments push out the plasma membrane so that there's more surface area, and so what does that have to do with absorption? If you have an organ that absorb material, like the small intestine absorbing nutrients, large intestine absorbing water, then the increase in the surface area will increase the ability to absorb material. But the microvilli itself, its function is not absorption. Right? So be real careful in biology, be real careful in this class that you don't get, you are very particular, specific about what you say, okay? So again, microvilli, the function of microvilli is to increase surface area. Why is increasing surface area? To increase the ability to absorb material, okay? Which is very different from later on when we talk about cilia, which is made up of or supported by microtubules, which has a different function. Okay, now we're going to go into talking about assembly and disassembly, polymerization versus depolymerization. Okay, so assembly, microfilament assembly. Assembly is a process. Okay, microfilament assembly is a process by which microfilaments are formed. The mechanism of assembly is polymerization. Right? So they're not synonymous. They're not the same thing. So assembly is a process. Polymerization is the mechanism. What is polymerization? Polymerization is the addition of one monomer at a time at the end, right? At the plus end in this case. So you're never going to have assembly where you have a chunk of monomer added all at once. It's always one at a time. Okay? So again, the mechanism is polymerization. Disassembly is the same, where disassembly is a process. So assembly and disassembly is a process. However, for disassembly, there are two mechanisms for disassembly. Okay? One is by a process called severing, and the other is by a process called depolymerization. So going back to talking about the capping protein, the capping protein prevents a polymerization or depolymerization, but it doesn't prevent severing. Does that make sense? So disassembly can still occur if you have a capping protein at the plus end, right? But disassembly in that case has to occur through severing and not depolymerization. Because again, the capping protein caps the plus end. There's no way, once the capping protein binds, there isn't an enzyme that removes the capping protein. So the only way to destabilize that microfilament, or the only way to induce disassembly, is by severing and not depolymerization. Okay? So what is severing and depolymerization? Depolymerization first. Depolymerization is the process by which a G-actin, a monomer, is removed one at a time from the end, right? One at a time. It shows here that it's removed from the minus end, right? But not at the plus end, and we'll go into more details about this in a minute, okay? But the idea about depolymerization is that a protein called cofilin, or you can pronounce it cofilin, I pronounce it cofilin. Cofilin is required. So cofilin is a protein that goes in and literally rip out one G-actin at a time, okay, from the end to carry out depolymerization. Cofilin can work at either the minus end or the plus end, and how does cofilin know which end to undergo depolymerization? We'll talk about in 2 minutes, okay? Severing is when you have a protein that goes right in the middle, not necessarily smack in the middle, but somewhere in the middle of the microfilament, and actually tear out, so it's not a protease, so it doesn't digest the protein, but it unbinds two G-actin that are binding, right? So basically cutting the microfilament right in the middle. Okay, that process is called severing, and the protein that catalyzes that reaction is called gelsulin or gelsolin, however you pronounce it. I'll pronounce it gelsulin. I have my own way of pronunciation. Okay, so far so good? So again, so if there were a cap here, that wouldn't affect severing, right? But if there were a cap, if it were at the plus end, but if there were a cap here, then it would affect the cofilin from removing the plus end. And if ARP were at this end, then cofilin wouldn't be able to induce depolymerization either. It has to be a free end, whether it's a plus end or minus end. Okay? Yes, sir.
Can you repeat that one more time?
Yes. So the ends have to be free before depolymerization can occur. Okay.
With the plus end after.
Right. So therefore, if there's a capping protein at the plus end, then depolymerization cannot occur. In fact, the capping protein prevents depolymerization, and that's how it does it.
It can't do it at the minus end if there's a cap on the plus end?
So looking at the end that needs to undergo depolymerization, if there is a protein that caps it, then there's no depolymerization. It doesn't affect the capping protein at the plus end has no effect on the minus end. So the minus end, then, if it has an ARP, then there's no depolymerization. But if there isn't, if it's free, then depolymerization can occur. So again, capping only affects the end where it occurs. Does that make sense? So the capping protein at the plus end has no effect on the minus end. ARP at the minus end has no effect on the plus end. And depolymerization only occurs if the end, whether it's the minus end or the plus end, were free.
So if there is a cap on the plus end, depolymerization will occur on the minus
Maybe.
end? Maybe.
Under certain conditions. Still have to be regulated. The only certainty is that the capping at the plus end has absolutely no role in what happens to the minus end. And the ARP at the minus end has absolutely no effect directly on the plus end, right? That's the only certainty that we know. Now, whether or not there's polymerization or depolymerization. At the other end, well, that depends on other type of regulation, which we're going to talk about right now. Yes, ma'am.
Is the cap always just on the plus end?
Right, the capping protein is on the plus end, and the minus end is ARP, which is used for nucleation, but can double as a capping protein in the minus end as well. But if you were to nucleate using formin, then there is no capping at the minus end, right? I know it's a lot of different regulation, and you need to know all of the above. Yes, ma'am.
With the severing, since polymerization can only happen one reactant at a time, it's the part that's cut off just degraded?
So let me, right, so that would get degraded. So then what is the difference between, following that, what is the most noticeable difference between disassembly by severing versus disassembly by depolymerization?
Go back to monomer.
One is faster, right? Much faster here, much slower here. So for instance, when we talk about migration, right, you want slow disassembly of microfilaments, so we'll talk about depolymerization, whereas when during cell division, when the cell has to be pinched, that has to be done really quickly. So disassembly of microfilament of the contractile ring will be through severing. Okay, so speed, basically. One is much faster in bulk, and like Deborah just asked, then that piece right there will just get disassembled, and then the G-actin will get reused. But then we'll have to pay attention to this stuff right here, another level of regulation. So before I go on, everybody okay with the regulation that we talked about so far? So nucleation is important, and then whether disassembly or assembly has to do with certain proteins that are present, not just that are present, but in which amount, right? In terms of thymosin and profilin, for instance. And then is cofilin around? If the minus end or plus end were free, but if you don't have cofilin around, guess what? You have no disassembly or no depolymerization, right? So and then without gelsilin, then there's no severing. Okay, so in addition to that, there's more, right? In addition to that, there's more. Yes, ma'am.
You sever the microfilament and it's in pieces, and it undergoes degradation. Is it done one at a time, like covalent, or
Right.
in
When this is broken down, it'll be broken down one at a time.
Oh.
Yes, yes, and chances are cofilin will help remove them as well. Okay. So can you see? And you should have the PowerPoint for this. You see that these each one of these circle is a G-actin, and then there's an orange, has an orange inner versus red, right? So the inside that is orange represent a G-actin that is bound by ATP. Red represent ADP. And remember what we said about GTP. GTP that is bound by an ADP cannot be used, not GTP. G-actin. What we said about G-actin. G-actin that is bound by an ADP cannot be used for polymerization. G-actin that is bound by an ATP can be used. Now here's the deal. So if an ATP bound G-actin, I have to be careful, is used and it adds here, what happened is that eventually that ATP will get hydrolyzed to become an ADP. Okay, so far so good. So if you keep adding, and what happened is that as the microfilament get elongated, get longer toward the plus end, right? The ATP that is bound to the G-actin toward the plus end, one at a time the ATP would get hydrolyzed. So then as you add more ATP bound G-actin here, the ATP would get would get hydrolyzed. So the ADP would follow suit. Does that make sense? Okay. The portion of the microfilament that still contain ATP where ATP hydrolysis hasn't caught up is called the ATP cap. So far so good? And you only have the ATP cap at the plus end. Why? Is because hydrolysis of ATP starts at the minus end, so you would never ever have an ATP cap at the minus end. So far so good? Yes, sir.
So the cap is just a region, it's not a molecule?
Right.
Okay.
So cap in this case is not a capping protein, it's a region,
Okay.
right? So it's just a region. So it's called the ATP cap. And that ATP cap is variable. What does the size of the ATP cap, how is that regulated? It's regulated by the speed of polymerization versus the speed of hydrolysis of ATP. If the cell is told to add a lot of G-actin at the plus end, then the hydrolysis of the ATP into ADP cannot catch up. Then you have a bigger ATP cap. If the rate of polymerization slows down, then the hydrolysis of ATP catches up, and then therefore ATP becomes ADP, and then therefore the ATP cap gets smaller. Can there be a case where you have the hydrolysis of ATP catching up with polymerization, whereas there's no ATP cap? Yes, right? That ATP cap can get negated. So far so good? No. What is not?
Sorry, could you just repeat?
Yes. So the rate of polymerization can be different from the rate of hydrolysis of ATP. If you have polymerization the rate being very high, right, then the rate of hydrolysis cannot catch up, therefore you have a big ATP cap. If the rate of polymerization slows down, right, and really the rate of hydrolysis of ATP is about the same always. So it depends on how high, how fast the rate of polymerization is. If the rate of polymerization slows down enough that hydrolysis of ATP catches up, you actually do have a case where you still have polymerization, but you have no ATP cap. Does that make sense so far? Okay. So then what is the function of an ATP cap? The function of an ATP cap is it is a condition that only favors polymerization and does not favor depolymerization. What does that have to do with severing? Nothing, right? Again, an ATP cap has nothing to do with severing, right? Severing can go right in the middle and just cut it off in the middle. Okay, so again, the ATP cap favors polymerization. So now let's put everything together. In order for polymerization to occur, you have to have a higher amount of profilin versus thymosin. You have to have a fairly high concentration of ATP-bound G-actin, and you have to have an ATP cap. And this is why polymerization and depolymerization occurs mainly at the plus end, right? You wouldn't have a lot of polymerization at the minus end. Why? Because there isn't an ATP cap there. Does that make sense? Okay, so those are the conditions. Again, you have to have an ATP cap to favor polymerization, and then you have to have enough ATP-bound G-actin, and then you have to have profilin binding, more profilin and thymosin. So all of the above concentration in a certain region as well. So if you look at the cell as a whole, that doesn't matter because we're looking at polymerization in a very particular region of the cell. Right? For instance, if you were to want to form a microvillus, then you better have all of these conditions right at the plasma membrane area where the microvillus has to form. Right? And if you have a lot of ATP-bound G-actin toward the nucleus, doesn't matter because that's not where the microfilaments are going to be formed. Okay, was there a hand back there?
Yeah, isn't ATP bound a product of polymerization?
The product of? No, it's the substrate of, not the product of. So ATP bound G actin is used for polymerization to get added to the plus end. Okay, yes sir.
Does ATP, I'm sorry, I'm a little confused, but the ATP cap is going to be on the negative end of
On the plus end
on
always.
the plus end, and as
Right.
polymerization
Because
catches
again-
up to it, we'll just break down.
Right. Because the explanation is, as you add ATP bound G actin toward the plus end, the ATP that is already bound to the G actin at the minus end will get hydrolyzed. So you will never get an ATP cap at the minus end because the ATP will get hydrolyzed. And the hydrolysis, the flow would be from the minus end toward the plus end. So ATP will not get hydrolyzed and skip around, right? So the next ATP that is hydrolyzed will be that one right there. And then the next one. So it's not going to skip all the way to the end. So again, the ATP cap is always at the plus end. Yes, ma'am.
So if you want to add the capping protein, then the capping protein has to be added to the ATP cap as well?
Well, the cap, right. So, okay, so interesting then. So for that, what happens is there's a capping protein here. You may or may not have an ATP cap, but it doesn't matter. The capping protein does not care. So if you have an ATP cap and then the capping protein occurs here, then no more polymerization, right, and no more depolymerization. So assembly and disassembly stops. But hydrolysis of ATP continues until there's no more ATP cap, right? So just answer that question, okay? So all the conditions have to be there, okay? So now let's go back to one slide and talk about this. So what happens if you have, let's say you have, here's a plus end. There's a plus end right here, and this is drawn weirdly. Actually, the way that it's drawn here, this is a plus end. So this is a plus end here, whether there's a cap or not, right? And let's say there's a cap. Okay, and gelsolin will sever here, and chances are there's no ATP cap here, right? Because the ATP had already been hydrolyzed into ADP. So guess what? Now this end is exposed. Okay, so now if this end is exposed, then you're certain that either assembly or disassembly will occur. Okay, in this condition where there's no ATP cap, then certainly the condition does not favor assembly.
Yeah.
But that doesn't mean that it favors disassembly either. So now, in this case, how do you regulate assembly versus disassembly? Now it has to do with the amount of thymosin versus profilin, the amount of G-actin, ATP-bound G-actin, but also the amount of cofilin, right? Because if you have a lot of profilin, but if you have a lot more cofilin, guess what? Cofilin will try to disassemble, carry out depolymerization. Does that make sense? So now it's just not comparing thymosin and profilin anymore. It's comparing thymosin, profilin, and cofilin. Think about it. There are different permutations. This is what your cell has to do. This is what your cell has to do, is regulate the expression of all of these things and ensure that the ends, whether it's the plus end or the minus end, favors either assembly or disassembly. Yes, ma'am.
So the side that has the cap and breaks away, you're saying that cap would prevent it from getting chewed up?
Well, the cap right here is important to stabilize. Well, what happened? Oh, I'm doing it. To stabilize the microfilament, but once that is severed and removed, that cap doesn't matter because this is a microfilament right here that remains intact, right? So this will just pretend that it's going to get recycled. So we don't care about that anymore. And the capping protein now is gone. So we don't need it anymore. Why is that? projector
Actually, this
itself.
water's moving too.
I'm sorry?
Computer.
Oh,
I wonder.
so somebody's dancing upstairs? I teach in Solano sometime, and they have a dancing class, and oh, they're the loudest people on earth. I actually have to ask the instructor to quiet down. Luckily, they were nice. So are we good with all this combinations of regulation? Okay. When we talk about microtubules, it's similar, but since I already talked about it, it should go over hopefully more smoothly. Okay. And then okay, yeah, okay, all right. And so the idea about severing, an example like I mentioned earlier was contractile ring here. And what happened is that the contractile ring is formed from microfilaments, but also because it's a contractile bundle, there are myosin filaments in between, and myosin and microfilament pull against each other, one another, to pull in the plasma membrane. But the microfilaments also have to be severed, okay? Because as you pull in, but if the size of the microfilaments were the same size, then the membrane can't be pulled in. Does that make sense? So you have to sever. And severing occurs instead of depolymerization because it occurs so much faster. Okay? So speed. So here, the myosin and actin filaments will pull against one another, okay, to pull in the plasma membrane, and at the same time, simultaneously, severing of the microfilament occurs. Okay? We'll come back to this idea again because it's an important idea and it's not easy to understand, so we'll come back at it again. Okay. A preview, migration, when cell cross or migrate, then polymerization occurs toward the end, toward the what we call the leading edge. And then the lagging or the trailing edge, there's depolymerization. Okay? And this is just a preview. That's all that you need to know for now. And we'll talk about the regulation later. So the idea for the last couple of slides is that this assembly is regulated differently. Different mechanisms are used for different functions. Right? So you don't always have disassembly by severing, or you don't always have disassembly by depolymerization. And the cell can't choose one or the other. The cell, if it needs to undergo severing, it must undergo severing. Depolymerization is not a good replacement mechanism, and vice versa. Okay? So that's all that the last two slides indicate. Okay, microfilaments. Should be slightly easier for microtubules because the regulation parallels with microfilaments. So therefore, let me stop for a few seconds and see if everybody is still okay and sane and alive for the next step. You okay? Okay. So microtubules are also dynamic. And the regulation of assembly and disassembly also have to be well regulated. Assembly is still through the mechanism of polymerization. Disassembly also has two choices, either severing or depolymerization. So everything is the same. So the difference would be in the actual molecules that regulate this. And there are slight differences, and we'll talk about the slight differences. First of all, nucleation. Nucleation is actually slightly more complex, okay? And there's that term MTOC again. So let me explain what MTOC is. An MTOC is a microtubule organizing center. Okay. Whereas for nucleation of microfilament, nucleation can either occur from a just free-floating aggregate of G-actin or a pre-existing microfilament, right? Neither of that applies to microtubule. Microtubule polymerization or nucleation must or requires an MTOC, a microtubule organizing center. If there isn't a microtubule organizing center, then there's no microtubules. But the reverse is also true, that if you can find a microtubule organizing center, chances are you will at some point have microtubules basically growing out of it. It's just almost like a pot in plants, right? If you have a pot, at some point you're going to put seeds in and the plant will grow. Basically, that's what it means. Okay? You've heard of a microtubule organizing center before. An example is a centrosome. You remember centrosome? Before cell division, there are two centrosomes that move toward the two ends, and then spindle fibers, elongation occurs. So a centrosome is an example of a microtubule or, organizing center. Something called a basal body is another example. And a basal body is important for the formation of cilia and flagella. Okay? And basal bodies are also important in terms of neurons, for instance, and very interesting. In terms of neurons, the presence or the location of the basal body indicates where either dendrites or axons are. So people who study, for instance, so some application to this, people who study spinal cord injury, right? So you have an accident and you have a carry-on or a cell body, but because of a spinal cord injury, your axons are severed, or your axon is severed from your neuron. And therefore, you might have the cell body, it's still working, it's not dead yet, but it can't send information, and that's why you can't move. Okay? So some of the research is to induce that cell body that no longer has an axon to produce an axon. How do you tell a neuron to produce an axon? By telling it to produce a basal body, a microtubule organizing center, and not only one, but where to put it, right? So you don't want a neuron here to tell your toe to wiggle, but yet the axon moves the other way. You don't want that, right? You want not only to tell the neuron to produce an axon, but where to produce an axon, and it has to do with the synthesis of a basal body in a certain area, right? So it's relevant. This type of information is very relevant to people's lives. So anyway, back to microtubule organizing center. So all microtubules are nucleated on microtubule organizing center. In addition to that, there is the equivalent of the ARP. There is not an equivalent of Foreman, but there's an equivalent of an ARP called the microtubulin ring complex, basically made up of gamma tubulin ring complex, basically made up of gamma tubulin. Remember, there are three types of tubulin: alpha, beta, and gamma. And alpha, beta are used to produce a microtubule. And so the gamma tubulin are not a part of the microtubule, but they are a part of this ring complex that catalyze or aid in nucleation of microtubules. And what happened is that the gamma tubulin then, gamma tubulin ring complex, actually anchor onto the microtubule organizing center. So these reddish rings here, that indicates the gamma tubulin ring complex, have to anchor onto the microtubule organizing center, and then the gamma tubulin then can carry out nucleation of microtubule. And so as many gamma tubulin ring complex as you see here, that's how many microtubules will radiate out from this centrosome. And if you have, let's say this is a basal body, then you would have all of the gamma tubulin ring complex lining up on one side, pushing out so that you can have a cilium or a flagellum or an axon or a dendrite. Okay? Now, after having said all of that, that's not really the full story. Because axon and dendrites formation are more complex than that. A lot more complex. Also, it's not the full story because we we haven't managed to find an MTOC in plant cells, and yet they still have microtubules. Okay, so the story that I just told you is a simplified story, and there are some complications that we don't understand yet. But in a nutshell, nucleation of microtubules requires an MTOC, and then it requires gamma-tubulin ring complex to adhere or anchor onto the MTOC before nucleation of microtubule can occur. However, very similar to microfilaments, polymerization occurs toward the plus end. Okay? So this is a little wrong. Should Curtis cross out Foreman? Gamma tubulin ring complex really is the equivalent ARP, not Foreman really. Okay. The equivalent of thymosin and both thymosin and profilin is Stathman. So Stathman does a similar job as both thymosin and profilin. Okay? So therefore it requires a little bit of explanation. So Stathman itself can be either in an unphosphorylated form as well as a phosphorylated form. Stathman that is unphosphorylated can bind to tubulin, the monomer. And so when Stathman is unphosphorylated, it binds to tubulin. Okay, and it inhibits assembly. So the unphosphorylated Stathman acts very much so like thymosin. So far so good? When Stathman is phosphorylated, Stathman no longer binds to tubulin. Tubulin, alpha and beta tubulins that are free from binding from Stathman can actually be used for polymerization without any additional binding. So slightly different, right? So let me repeat the whole thing. So let me repeat it, but the reverse, okay? So free alpha and beta tubulin without requiring to be bound by anything else can be used for polymerization of microtubule, right? And Stathman, if Stathman were unphosphorylated, then Stathman can bind to alpha and beta tubulin to prevent alpha and beta tubulin from being used for polymerization, but only in the unphosphorylated form. So in order to negate the activity of Stathman in inhibiting polymerization, then Stathman needs to be phosphorylated. So therefore, the level of regulation is to regulate whether Stathman is phosphorylated or not in the expression of, well, you still need Stathman to be phosphorylated, expressed, but not in comparing to anything else, right? So the comparison will be in terms of how much unphosphorylated Stathman is versus how much phosphorylated Stathman is. And if you have more unphosphorylated Stathman, then no polymerization. If you have more phosphorylated Stathman, then the alpha and beta tubulin are free to be used for polymerization. Yes? No? Repeat something?
Can the Stathman, like the difference between the phosphorylated and the unphosphorylated, can that be regulated by covalent modification?
Yes. Well, that is covalent modification. Stathman itself is covalently modified. Oh, but other covalent modification? I don't know. So basically, what people in the field who study this would actually look at the expression and activation of the enzyme, the kinase that phosphorylates Stathman, and also the phosphatase. So the idea is the fight is basically between the kinase and phosphatase that would regulate Staffman, right? If the phosphatase wins out, then Staffman would be unphosphorylated and therefore no polymerization. If the kinase wins out, then Staffman would be phosphorylated and then you have polymerization. So it's multiple steps, right? At first it's the fight between kinase and phosphatase, and then it would be the comparison between phosphorylated and unphosphorylated Staffman, and then it'll be whether you have polymerization, all of which also involves how much alpha and beta tubulin are there. And remember also that alpha beta tubulins must be GTP bound to be used. In the GDP bound form, alpha and beta tubulin cannot be used for polymerization. So lots and lots of regulation, and you need to know all of the above. So this is a fantastic time to ask questions. If you don't understand what I said, or if you just need a few minutes to digest, or if you need a few days to digest and then come back and ask questions on Thursday, that's fine too. But I need you to understand all those levels of regulation. And there's more, by the way. Yes, ma'am.
Can you go over the phosphorylation again?
So Staffman, when unphosphorylated, can actually bind to tubulin. And when Staffman binds to tubulin, as it states here, Staffman binding tubulin will inhibit assembly. Right? So tubulin, alpha and beta tubulins that are bound by Staffman cannot be used for polymerization. So in order to be used for polymerization, tubulins have to be free from binding by Staffman, also have to be bound by a GTP, right? And in that case, then free GTP bound alpha and beta tubulin can be used for polymerization. How do you free the tubulin from Staffman? By phosphorylating Staffman. How do you regulate the phosphorylation of Staffman? By regulating the kinase that phosphorylates Staffman. Either by increasing or decreasing expression, and or by regulating the activation and inactivation of the particular kinase. Yes, sir?
Is it safe to say then that Staffman is in essence acting as the cap? Because its presence would inhibit the elongation of the tubulin, correct?
No, but it doesn't act as a cap. It's the unphosphorylated form actually acts like thymosin,
So it's still signaling, the kinase
binding to the subunit to prevent polymerization, but not a cap.
microtubules.
So this is irregardless. It doesn't matter what happens at the plus end of the microtubule. This is regulating whether or not the tubulin will be used for polymerization or not. Okay. It does have something to do with the and we'll look at that. It actually has the same activity as cofilin as well. So I'm trying to do this slowly to add a little bit of pain at a time instead of go all out. But I want if you would let me know if you understand each step, or if I'm just talking way too much and this is easy stuff, that's fine too. But just let me know how I'm doing so that I can help you understand the stuff, right? At the end of the day, you and not me have to take the exam, so this is all for you. So far so good. Are we okay with at least the regulation by Staffman? Right. So pretty picture. So speaking of capping protein, there is not an equivalent. There's something similar to a capping protein, but not the same. Okay. So there's no such thing as a capping protein for microtubule. So therefore, the ends are never capped. There are microtubule associated proteins that act similarly to the capping protein by actually binding toward the plus end of the microtubule. Right? So binding to the side toward the plus end. So let's say the microtubule is this long. Then these proteins, either MAP2 or tau, doesn't matter if you either one. You just know that if I mention MAP2 and tau, you know that these are proteins that bind to the side of the microtubule toward the plus end. And what do? They have, again, a similar function as capping protein, but not exactly the same. MAP2 and tau, these proteins stabilize the microtubules. What that means is that these proteins will inhibit depolymerization. Not disassembly as a whole, because again, severing can still occur, but MAP2 and tau stabilize the microtubules by inhibiting, literally inhibiting depolymerization. Okay. However, since the end is not capped, therefore, polymerization may still occur, but of course depends on other things, right? So again, there's no such thing as a capping protein. The closest that we come to a capping protein for microtubules are these microtubule-associated proteins MAP2 and tau, where they bind to the side of the microtubules toward the plus end. Okay? And MAP2 and tau, what they do is they prevent depolymerization, but they do not prevent polymerization. But don't say that they encourage or favor polymerization either, because favoring polymerization has to do with the GTP cap. So far, so good?
So they inhibit polymerization?
No, they inhibit depolymerization, and they don't do anything to polymerization. Okay? Polymerization may still occur, but there are other regulatory processes that must be in place. So far, so good? So what are those, let's see. Let me do this and then come back to this. So what are those regulatory additional regulation that must occur? So these purple thing, let's say on this side, represents MAP2 and tau, right? So they stabilize the microtubule and therefore prevent depolymerization. But in order for polymerization to occur, you have to have the following. Number one, there has to be a GTP cap. And the GTP cap is very much so analogous to the ATP cap, where, remember, tubulins must be bound by GTP before it's used for polymerization. But as GTP bound tubulins are added to the plus end, the GTP that are bound to the tubulin already in the microtubules get hydrolyzed into GDP. And so the hydrolysis of GDP will follow the addition of tubulin. And if the rate of polymerization is high enough where hydrolysis of GTP hasn't caught up, then you will have a GTP cap instead of an ATP cap, right? Same difference. And so again, in order, if MAP2 or tau will bind to the microtubules toward the plus end, and to prevent depolymerization, however, for polymerization to occur, there should, it's not absolutely, it's not an absolute requirement, but if there were a GTP cap, then that would encourage polymerization. Okay? But other things that are required would be the fact that alpha and beta tubulins must be bound to GTP, the free form. Also, that Stathman must be phosphorylated. Right? So if there's a GTP cap, but if you have a high enough amount of unphosphorylated Stathman that would bind and sequester or gobble up all of the alpha and beta tubulin, guess what? You don't have any monomers to be used for polymerization, right? So in addition to having a GTP cap that would favor or encourage polymerization, you still need to have the alpha and beta tubulin bound by GTP and free from binding of Stathman. And in order to free the alpha and beta tubulins from binding by Stathman must be phosphorylated. In order to phosphorylate Stathman, then the kinase that phosphorylates Stathman must be activated. Right. If you have a cap, if you have a MAP2 and tau that binds, but the condition is not, does not, all the conditions do not favor polymerization, then guess what? Now the microtubule is stable because depolymerization can occur. And the condition does not favor polymerization, so therefore the microtubules stay in place. Okay, yes, sir.
So with the GTP, polymerization is encouraged, but if
Favors.
or is
Favors
favored,
it, right.
but if a MAC2 or the tau protein is attached, it'll stabilize it and it will not go through polymerization?
No. So those are two different things. A GTP cap is different, independent of whether or not MAC2 and tau bind. Let's say without MAC2 and tau binding, right, and you have a GTP cap, then that favors polymerization anyway. Okay, and depolymerization will only occur once the GTP cap is gone. Now it has to go the other way, right? However, if you have MAC2 and or tau bind, right, then regardless of whether you have the GTP cap, depolymerization won't occur. So it's either polymerization or nothing at all, is what that means. So that means that MAC2 and tau are very important, right? That answers your question, I think.
Yes.
Okay, are we good? All right. So for depolymerization, again, if whatever condition it is that favors depolymerization occurs, so they're analogous to covalent that rips out a ADP bound G actin one at a time, then depolymerization of microtubules can be carried out by either a factor called a catastrophic factor, a catastrophe factor. There are different ones. Kinesin 13 is one, and guess what? Unphosphorylated Staffman is also a member of the catastrophe factor, as indicated here. Okay, so Staffman has more functions than when we saw for thymosin and prophylactin. Okay, so it has multiple functions. So Staffman, in other words, Staffman can bind to tubulin that are either free or already bound to the microtubule, whereas thymosin and profilin will only bind to free G-actin. Right? Staffman can bind to either free or bound form of tubulin, either the GTP or the GDP bound form as well. So Staffman has its binding site. It has the binding site of Staffman is a lot more flexible. That's why it has more functions. Okay, and then the equivalent of gelsilin is I always like this. There are a number of proteins as well, and one is catenin. And any martial arts buff would know what that means, right? Anybody know what catenin refers to? That samurai sword. It's called a catenin, right? So that's the name. That's where the name came from. Is that spastin is another member of the of enzyme that carry out severing. Okay. So we're good. We're good. So the comparison between assembly and disassembly of microfilament versus microtubules, there are a lot of. Most of the mechanisms are similar, and then there are differences throughout. Okay, so just like an example of how all of this play out, and again, just like microfilaments where severing and polymerization, disassembly by severing versus disassembly by polymerization occur at different times, at different locations, and it's not a choice, right? If a process, if a microtubule or microfilament must be severed, they must be severed. Depolymerization cannot replace it. And if they need to be depolymerized, then severing will not who would not replace it. Okay, so let me go through one scenario for you. So during the prophase of mitosis, sister chromatids are pushed toward the middle, right, toward the metaphase plate or spindle equator, and then that means that polymerization of microtubules has to occur. And then during anaphase, the sister chromatids are pulled apart, and that is carried out by depolymerization to pull them apart. So let me see. Okay, what I'm looking for is how much detail I give you, and I won't give you much detail right now. We'll look at this again with a lot more detail later on. But for now, basically, what you're looking at is both depolymerization and severing occurs during the process of anaphase, where at the beginning, these sister chromatids are pulled apart due to depolymerization. So catastrophe factors like kinesin 13 and Staffman will remove one tubulin at a time, right, to allow for the sister chromatids to be pulled toward each pole. Once they get close enough to each pole, the mechanism decided that, well, enough is enough. This is taking way too long, and severing occurs. So and why not before here? Because if severing occurred here, these sister chromatids might float around and they might be moved. They might diffuse literally toward the wrong pole. But if they're pulled close enough to the respective pole, the idea is that now they're not going to have time to diffuse toward the other pole, because around this time the nuclear membrane is being formed as well, right? So severing occurs. So during anaphase of mitosis, at the beginning depolymerization of these microtubules occur, and then toward the end of anaphase and leading into telophase, then severing of microtubules occur. Okay? So that gives you an idea. We're going to look at this in greater detail later on. For the next set of exam, I think. I don't think it's this set of exam. Okay. So are we good with microtubules, microfilaments? Yes, ma'am.
Do you want us to specifically know some of the catastrophe factors?
Just know that they're catastrophe factors. I'll just use the term catastrophe factor. Good enough, right? Intermediate filaments, the regulation of intermediate filaments. We're not going to, we'll have time, there's plenty of time. We'll get done with intermediate filament. It's actually not as complex as regulation of microfilament and microtubule assembly and disassembly, but a lot more complex than what I will introduce. Okay, so in general, you notice that again, intermediate filaments are very stable. So therefore the assembly, the assembly is going to be pretty slow as well, right? So you take two subunits, you twist them into what is called a coil dimer, and then you take two of those dimers, you slam them side by side, and then you take these tetramer and then you lay them end to end, and then when end to end you take 8 of these and then you wrap them up in an intermediate filament. So this process is very slow, okay? And the requirement, there aren't too many associated proteins that are required, and most of the regulation occurs through covalent modification, either phosphorylation or glycosylation. Okay. We'll only look at an example where phosphorylation occurs, and we'll only look at the example for nuclear scaffolds. So again, regulation of assembly and disassembly, both assembly and disassembly for intermediate filaments involve mainly covalent modification. And the two types of covalent modifications are mainly phosphorylation and glycosylation. However, we're only going to pay attention to phosphorylation. And some of you are already thinking, well, glycosylation, but it's in the cytosol, how can it be? Remember, this is the insoluble. This is the insoluble part of the cytoplasm, so a lot of the glycosylated portions are actually going to get buried anyway, okay? But we'll just pay attention to phosphorylation anyway, okay? So roughly, here's how it works. So going back to mitosis again, okay? So the nuclear scaffold, we'll use that in an example. Lamin. Lamin, when lamin is in the unphosphorylated form, unphosphorylated form, lamin have affinity for one another. So in the unphosphorylated form, lamin almost spontaneously will form a nuclear scaffold. Okay? And during mitosis where the nuclear scaffold does what? It actually form and support the shape of the nucleus because it basically props up the nuclear membrane, right? So during mitosis, during the prophase of mitosis, what's one of the earliest events? The disintegration of the nuclear membrane, right? So how are nuclear membranes, how do they break down or disintegrate it? By no longer having a nuclear scaffold. So if the nuclear scaffold breaks down, then the nuclear membrane breaks down. Right? Because again, the reason why the nuclear membrane is formed and the nucleus is formed is because of the intermediate filaments of the nuclear scaffold. So if the nuclear scaffold is broken down, then there's no more nuclear membrane. How can a cell induce the breakdown of the nuclear scaffold is by phosphorylating lamin. Okay, so lamin in the unphosphorylated form can be used to form the nuclear scaffold. But if there's a kinase that goes in and phosphorylate the lamin that are already in the nuclear scaffold, then phosphorylated lamins no longer have affinity for one another, and that causes a breakdown. So that causes disassembly of the nuclear scaffold. So it's almost, not quite, but almost as simple as phosphorylation versus dephosphorylation. Okay? But in this case, dephosphorylation then allows lamin to be unphosphorylated. Therefore, dephosphorylation of lamin actually encourages or favors assembly, versus phosphorylation of lamin actually favors disassembly. And phosphorylation of lamin can be done on either free lamin or lamin that's already in the nuclear scaffold. So in addition to phosphorylating lamin that's already in the nuclear scaffold to allow for the disassembly of the nuclear scaffold, if you have free lamin around, then they will also be phosphorylated so that they will not be used for polymerization. Right? So that we're going to find out that whenever a cell receives a signal, it doesn't just receive a signal to do something. It also receives the signal not to do the opposite. For instance, a cell that is told to die will also be told at the same time to live. I know that sounds like wasteful, right? So if you're going to be told to die, of course, by definition, you're going to be told not to live, but unfortunately, that's not how the cell works. Okay? So if a cell were told to live, then it will also receive the signal not to die. So cells always receive multiple signals to tell it to do something, but not to do the opposite. So the same thing here is that phosphorylating lamin basically is a signal to tell the cell to undergo disassembly of the intermediate filaments, but also will tell the cell not to undergo polymerization by phosphorylating free lamin as well. Right? Good. Let's stop there. You look tired, and I know I'm gone, so have a